Hi, everyone. Welcome to our second annual Finish Line event. We're so excited to be here tonight and have you all join us. This is a big event for us and we're very proud of it. It's a great night because it gives us a chance to celebrate what we do best, which is our research. And it gives us a great opportunity to share some updates and what the foundation has been up to. It also sort of signals the ending of our 1-month fundraiser, the Steps Move Science event, and it gives us an opportunity to thank all the participants as well. So tonight we have a great lineup for you guys. I'll give a chance to introduce our board members and then give you an opportunity to hear from each of our researchers. You'll also hear a bit of an overview of our scientific advisory panel, which is great. Also gives us a chance to celebrate the participants in our event. So thank you all for being here. I'd like to start by sort of giving you a bit of an introduction of our board members, give you a chance to meet them virtually, so to speak. Myself, I'm Jennifer Iannuzzi. I'm the president and co-founder of the SMS Research Foundation. We also have John Miller, who is our vice president, Dan Longman, who is our treasurer, Chris Iannuzzi, my husband, who heads up our SAP. And then I'm excited to share that we have two brand new board members who just joined us, Jeremy Hulot from Canada and Jennifer Sauer from Texas join us recently. I am thrilled to be working with them. The little I've had with them, I'm very excited. Their enthusiasm is fantastic. Jeremy has already done some fundraisers for us, as well as Jennifer, and Jennifer is full of exciting creative ideas, and I look forward to them. So without further ado, I'm going to hand it over to Chris, who heads up our SAP, and he's going to share a bit about that.
Welcome. Thank you. I'm really pleased to be here and excited about tonight. What we'd like to start with is an animation video that we created. We know a lot of families with SMS are on this webinar, and certainly they know the definition of what SMS is. But often when they try to explain it to friends or family or professionals that work with their children, it can be difficult to really discuss the complexities of a microdeletion syndrome. So we made this simple video that we hope will be helpful to you. So Terrell, if you don't mind showing the video.
Smith-Magenis syndrome, or SMS, is a complex genetic disorder. It is referred to as a microdeletion syndrome because a small part of DNA is either missing or not functioning properly. Every cell in the human body contains 46 chromosomes, 23 pairs. A chromosome is a long piece of DNA that contains the genetic information of an individual. This genetic information is sectioned into genes that are unique sequences derived from parents and transferred to offspring. Genes code for important proteins that play a critical role in human cell regulation, facilitate proper growth and development, and give us our unique characteristics. For those with SMS, one copy of the RAI1 or retinoic acid-induced 1 gene is missing, or one copy is mutated, such that it is rendered non-functional. Therefore, this gene is not functioning at the proper level. This has detrimental effects on all the proteins in the cell that it would normally interact with to do its job. The brain is a key tissue that requires proper RAI1 protein levels for growth and development. The lack of sufficient RAI1 production leads to significant clinical and behavioral issues, which can include self-injury, aggression, impulsivity, emotional dysregulation, obesity, sleep disturbances, and cognitive impairment. Unfortunately, this constellation of features makes it extremely challenging for individuals with SMS to become fully independent, and they typically require a lifetime of supportive care. The SMS Research Foundation is dedicated to finding unique and innovative ways to improve the lives of individuals with Smith-Magenis syndrome. Our research is based on 3 approaches. 1. Replace the bad copy of the RAI1 gene with a good copy using gene therapy. 2. Stimulate overproduction in the good copy to make up for the bad copy using CRISPR technology. 3. Find or develop a drug that can promote the production of RAI1. If RAI1 can be replaced and properly regulated, it is possible that some of the most difficult features and cognitive delays may be improved. Supporting this kind of research will lead to the most effective treatments for people with Smith-Magenis syndrome.
Thank you, Terrell. So We would really like to recognize our top sponsor tonight, which is Vanda Pharmaceuticals. They have a drug called Hetlioz. It's the first and only FDA-approved treatment for nighttime sleep disturbances and SMS. We'd like to show their commercial because we really appreciate all the support they gave us this evening.
1 in 15,000 people is born with Smith-Magenis syndrome, also known as SMS. SMS is a complex developmental disorder linked to chromosome 17, associated with severe sleep-wake cycle disturbance. People with SMS may be awake when it's time to sleep, and asleep when it's time to be awake. Visit smithmagenis.info or call 833-433-4331 to learn more about sleep disturbances and SMS and find out what options may be available to you.
Perfect. Thank you, Terrell. If you need more information about that drug, you can certainly contact us or you can go to their website. I think you've got some additional sponsors, Terrell. Great. So that the Vanda is our ultra sponsor. And then Harvey is a walker sponsor, and our stroller sponsors are STP and Living Special Needs. And I want to thank all of them for supporting this event. Now, the main purpose of this evening is to give our community and our supporters the opportunity to hear directly from our researchers and to really get an understanding of the work that our foundation supports. In order to be good stewards of the money we receive and to maximize the potential, all of our research decisions are overseen by a scientific advisory panel. You can see all of our members there in front of you. This is a group of very highly accomplished scientists that are dedicated to basic science research. I encourage you to go to our website, smsresearchfoundation.org, reviews our process in general, and also you can look more thoroughly into the biographies of these great scientists. I will tell you that the linchpin and the person who will lead us in the scientific program this evening is Luca Parazzi. Luca is a friend. He's also the father of Valentina, a little girl with SMS. He is a physician scientist. He has a PhD in immunology. He was the head of development for rare diseases and gene therapy at Sanofi Genzyme, and he's currently a senior vice president and the head of development and safety at Alexion AstraZeneca Rare Disease. He's a senior member of our SAP and really an invaluable research for our scientific program. It's really a privilege to introduce him. And Luca, thank you for taking us through the scientific portion of our program. And before I hand it off to you, if I could just ask Terrell to please make Jess Carden a panelist. Thank you, Luca.
Thank you very much, Chris. It's an honor to be here today. And thank you for you and Jen and everybody else on the board for all your tremendous efforts to make this happen. The reality is that you guys are the architect of all of this. It's a pleasure for me to be part of the scientific advisory panel. As a matter of fact, the scientific advisory panel is a group of scientists and also friends. We meet periodically every couple of months. We review the progress of the research. We keep an eye on that. We provide suggestions to our researchers. We have very good and solid scientific discussions. And I'm very pleased for the quality of the research and all the discussion we've been having in the last year. It's been absolutely impressive to see the tremendous efforts and improvement in the efforts we're doing in Smith-Magenis syndrome. Now, research will allow you to pursue your interest, to learn something new, to hone your problem-solving skills, and also challenge yourself in many new ways. Research is what propels humanity forward. And for a rare disease like SMS, in fact, for any rare disease, I would say, it becomes really essential. Very little is known about the underlying causes of rare diseases. Very little is known about the physiopathological mechanism of our diseases. And sometimes we don't even know what is the most effective approach to study the disease. Sometimes we don't have an animal model to study the disease. So we have to start from the ABC, developing a model first, and then be able to do our own experiments. So it's a very long journey and a very complex journey. And obviously, when you're a parent and you're directly involved in a rare disease, it becomes a personal matter, like many of us on the call. We started these journeys years ago, and I am very proud of the progress that we have made so far to unlock some of the basic understanding of SMS, putting us closer to therapeutic interventions. All this progress would not have been possible without your generosity, without your support, without all the efforts of our donors, the fundraisers, People are spending their time and their lives collecting money and supporting research. And of course, the contribution of our scientific advisors who are doing that obviously out of free of charge and generously dedicating their time to be able to support our research. And of course, generosity of the researchers themselves that dedicate their lives for the cause of SMS. So I would like to thank you all for all of this. Thank you for believing in our mission and eventually for improving the lives of our wonderful kids with SMS. Now, keeping that in mind, one of the main goals of our scientific advisory panel is to establish research priorities, which of course are extensively discussed and approved with the board. Now, our research priorities are summarized here. We would like to gain interest and understanding of RAI and its role in brain development. And that, of course, is the base of the research. First of all, we want to understand the underlying pathological mechanism, as I mentioned previously, understand how the gene works. And when we get to that, the next step is development of novel therapeutic approaches with the goal to reestablish the normal expression of RAI and eventually having a therapeutic benefit for all our kids and our patients with SMS, and honestly also for the families. We all know that this disease is not only our children's disease, it's a family matter. It's something that impacts the entire family and the children, the teachers, and entire community around them. So it's not just about them, it's about all of us, as a matter of fact. Now, keeping that in mind, the next step, there is just a slide that summarizes the annual grant program process. The annual grant program is done to fund novel scientific research, either from established researchers who have been working for a while, also from new researchers. So we're very glad to have some new research coming up since last year, and we think that the scientific efforts we've seen through this new research is going to bring our efforts to the next level. Obviously, there's a lot of efforts from fundraising to fund and to find the money, and then there's an 81,000 grants per year, and we also researchers can apply for additional funds for the research as well. Usually what we do, we receive a concept paper with a proposal. We receive many of them. We analyze, review carefully all of them, and then the scientific advisory panel comes to a conclusion on the few programs we believe are the most interesting one, and we're going to fund for the year. That's how we came up with the proposal that we use tonight. So without further ado, I would like to start the scientific discussion tonight. I'm going to introduce each of our researchers one by one. I will ask them to share the screen and their presentation. I will also ask the panelists to please stay online up to the end of the presentations, because we have a Q&A session at the end, which I'm going to moderate. So I'm going to look at the questions we receive and distribute them to our researchers as well. With that, I will start with Professor Wei, understand the function of RAI and develop new therapy options using different experimental models. So the project that we're going to discuss today has a high translational impact and provide us a unique path forward for future development for therapeutic intervention to be able to normalize RAI levels and improve the quality of life of our patients with SMS. And with that, I would like Wei Zhan start his presentation.
Okay.
Thank you so much.
Okay, so my name is Wei Sheng Huang. So I want to tell you a little bit about the research of my lab. So my lab is entirely focused on Smith-Magenin syndrome, and we are focusing on a few different projects, including the neurofunction of RL1, as well as developing different kind of therapies for SMS using mouse models. So I want to tell you first about how do we understand the basic neurobiology of RL1. We are first focused on brain activity because SMS patients commonly have abnormal EEG brain activity patterns, and some patients have clinical seizures. And this is a problem because seizure is a hyperactivity of the brain, if you will, and too much activity will maybe sometimes damage your brain, and then it will impair the quality of life. And one of the problems to understand how RL1 regulates brain activity is that, at least in mice, and also that's true in human, RL1 is expressed throughout the brain. is very widely expressed. So how do we identify brain regions important to seizures, right? Like what brain region is responsible when you have RL1 loss to drive neural hyperactivity? So to do this, we use a technique called brain clearing. Basically, as you can see here, this is the before and after clearing. We can basically clear the whole mouse brain to remove all the unnecessary lipids and do whole brain imaging and immunostaining. So we clear the mouse brain, and then we stain for one protein called FOS, which is a marker for brain activity. So if a brain region has too much FOS protein, that means that that brain region has a lot of activity. And after whole brain imaging, because the mouse brain is now clear, we can look at the whole brain. We can identify brain regions with too much FOS signals. So here's an example of the FOS signal. So I want to focus on this region of the mouse brain called hippocampus. And we found that in RL1 mutant mice, there are too much FOS protein if you use a canic acid treatment, which is a drug to increase brain activity. This brain region will show too much FOS activity. So that suggests that this brain region could be hyper-excitable in the SMS mice. So to look at this problem in another way, we put electrodes in the mouse brain and record the brain activity, and we can see that our mutant mice, they spend more time in seizure. So this is a light mouse, right? So this is another evidence suggesting that losing RL1 in the hippocampus, so the electrodes are actually put into the hippocampus, have too much activity. And then we can also do another way by taking the cells out of the mouse. We can prepare acute brain slices and basically use a pipette to listen to the neurons talk, if you will. So this graph shows you that in the control mice, there's some activity in the neurons coming from the surrounding neurons, the neighbors. And in RL1 knockout mice, the neighbors talks a lot, right? There's more activity from the neighbors. And how about RL mutant neurons themselves in the dentate gyrus, hippocampal dentate gyrus neurons? We can see that the RL mutant neuron themselves also talks a lot, very frequently. So this kind of hyperactivity could be contributing to the susceptibility to seizure in SMS mice as well as patients. So what happened to these neurons, right? So we analyzed gene expression and found that there are upregulation and downregulation of some ion channel proteins. These are the proteins that allow the flow of ions in and out of the cells. So for example, too much of this protein will allow too much calcium ion coming into the cells and potentially make the cells more excitable. So we tested by adding a drug called NNC55 to inhibit this ion channel. And as a result, we can see that when the RL mutant neuron, they talk too much, but if you apply this drug to block this ion channel to reduce the activity of this channel, then the neurons will become quiet down. So we are looking into this and to see if we can further understand what's going on here. But this is just one example of how we identify the important brain regions in the SMS mouse brain and then understand what the molecular and cellular mechanism that potentially contributes to seizure in the mice. So now to gene therapy project. As you can remember from the video, every patient has a good copy of RL1, right? So another way is how do we increase the expression of this good copy to treat SMS? So we're using a technology called CRISPR activation system. Basically, they have two components. One component is the RNA component. Basically, this molecule here will direct another component, the protein component, to the RL1 gene. So this is the remaining good RNA copy. We want to find out a way to enhance the expression of this copy. So with the protein component and the RNA component together, this protein can activate the expression of the nearby gene. In this case, because this RNA is located upstream of RL1, so the protein can activate RL1. So this will be the concept of this design. So basically now we found we identified different RNA that can bind to the upstream of RL1 gene, trying to increase RL1 expression. And we found a sequence, RNA SG2, we found that you can increase actually RNA expression by 2-fold. So this is promising. And then we make adeno-associated virus, AAV, which is the gold standard for gene therapy right now. So we replace all the toxic genes and then put in the necessary component, the protein component and the RNA component I talk about. We made a virus out of it, and then we treated neurons with this AAV virus to see if we can increase RL1 level or not. So this is a wild type mice, right? We use it as the 1. And the SMS mice, the neuron have few lower RNA expression. And we found that with the treatment of this AAV, we can increase RL1 expression level. So that is very encouraging. Then we injected this virus, the treatment virus, into mice. So we select a brain region in the hypothalamus called PVH. So this brain region, in our recent work, we found that it's important to mediate obesity in the SMS mice. So we thought this would be a good place to start. So we basically generated wild-type mice with control virus, SMS mice with control virus, and SMS mice with the treatment. So the purple one is a treatment group. One of the features of SMS mice is they have a lot of repetitive behaviors, and you can see here in red. But after treatment, you can decrease the repetitive behavior, as you can see here in purple. Another feature of SMS mice is the increased body weight, right? Compared to control in black, SMS mice have eventually gain body weight, as you can see in red. And after the virus treatment, we found that although the mice eventually still gain weight, their body weight is much lower than the SMS mice receive control virus treatment, and the onset of their obesity is also much delayed. So this suggesting that the good copy of Rl1 has some effect. And to remind you, we treated the mice at about postnatal 3 weeks of age. So it is like an early adolescent, equivalent to humans. So to summarize the second part of our project, I told you we used a novel CRISPR AAV system that have a protein component and the RNA component to increase RL1 level. And we can inject the virus that encodes this component into the mouse brain of SMS mice to increase RNA expression and then partially rescue some of the obesity and stereotypic repetitive behavior. So we can understand the remaining good copy of RL1 can modify, but if you increase the expression, it can modify disease progression. So the final part of the project, also the part I'm more excited about, is developing a BDNF therapy for SMS. So RL1, our previous work found that it's a protein that binds to DNA and then up-regulate or down-regulate a lot of different target genes. And one of the important target genes of RL1 is called brain-derived neurotrophic factor, or BDNF. And SMS, because they have fewer RL1, so it have decreased BDNF expression in the hypothalamus region. So what BDNF does is that it's a secreted molecule. It binds to the downstream receptor of surrounding cells to activate downstream pathway that promotes neuronal health. So if you don't have this BDNF, you have fewer of this BDNF, you have fewer of these signaling pathways. So as a result, neurons might not be as healthy. So our first experiment, we figured, why don't we put human BDNF back into the mice? So this is the human BDNF transgenic mice that basically express human BDNF from early development. So we want to ask, what if the SMS mice develop with the normal amount of BDNF? So you can see here in blue is a treatment group. Again, SMS mice is the red one. BDNF level is increased with the human BDNF expression. And then the obesity you see in the red group, the SMS group, is gone in the SMS mice treated basically genetically expressed human BDNF. And the body weight remains similar to control mice, even when they are very old. So that means that if you have BDNF expression early in life, you can prevent the development of obesity. So HDL, LDL, and leptin are some of the protein and lipid that is associated with obesity. And they are dramatically increased in SMS mice, but they're also decreased when you have human BDNF over expression. So these are the section of the adipocyte of fat cells. So in SMS mice, the fat cells are quite big and large. They accumulate a lot. But if you have a BDNF treatment, again, they look very similar to control mice, suggesting that the size of the adipocyte can be decreased by this genetically expressed human BDNF in mice. So another defect we can see in SMS mice is that they are more shy, if you will. So there's an assay, if you put two mice on the different end of the tube and you let them meet in the middle. And what happens most of the time is in control mice, they will push forward. They are very interested in this kind of social engagement to the extent that SMS mouse usually will just retreat and then leave, sort of losing the match. And what happens here is SMS mice will basically retreat. They are more shy. They leave the tube 95% of the time. But with human BDNF expression, they behave more like a control mice. So they win 50-50% of the time. And when they encounter other SMS mice, they become less shy, right? So they stay in the tube more. They engage in this kind of face-to-face social engagement more than the SMS mice, suggesting that expressing of human BDNF can also modify disease progression in terms of social interaction. So this is all sound very nice, but this is a genetically expressed molecule from early development. But can we figure out a way to use pharmacological approach and to increase BDNF signaling? And especially we are interested in adult mice, because I think in treating adult mice is maybe having more impact in the human patient population. So just a reminder, right? BDNF is a secreted molecule. It has to bind to TrkB receptor to enhance downstream signaling. And we found a drug called LM22A4. It is a small molecule that mimics the BDNF domain. So basically, it can also enhance the TrkB signaling pathway. It has been shown to have some potential in other disease. And we are using this drug to see if we can reverse or delay some of the symptom progression in adult SMS mice. So this project will be the main focus of our lab next year. But we do have some interesting preliminary results showing here. So we have very few number of mice. So here in orange is a control mice treated with saline daily, and we have 3 experimental SMS mice treated with this drug I just talked about, also daily. So we have 3 of them. And when we treat them at 8 weeks of age, so they are considered adult. So basically, we can see that the body weight of these mice treated with drug decreased dramatically compared to control. And we isolated their fat from liver or other organs. We found that in the control SMS in gray, they have increased fat mass, but with the drug, we can sort of decrease these fat mass. So the goal for next year is to use this drug and then analyze if this drug can rescue obesity and social interaction and repetitive behavior in SMS mice. So to summarize my talk, I show you we studied the basic neurofunction of RL1, and we identified that RL1 loss causes a hyperexcitability of hippocampal neuron in the brain that could potentially facilitate seizure. And we also have a drug that can decrease the hyperexcitability of these neurons. I show you that using CRISPR activation gene therapy, we can increase RL1 level in mice and then partially mitigate some of the disease feature. We show you that the genetic expression of human BDNF can prevent the development of obesity and social interaction defect. And finally, for next year, we will test in pharmacological treatment targeting the BDNF pathway to enhance it, can reverse or halt the disease progression in adult SMS mice. So I'd like to thank my whole lab, and then I'd like to thank Sms Research Foundation for supporting our research. Right, I started my lab in the middle of the pandemic, so it's quite difficult. It is actually impossible without the support of Sms Research Foundation. So we are really grateful for that. If you have any question, you can email me. Here is my email address. Thank you.
Excellent. Excellent. Thank you, Wei Shang. This is I would suggest to actually start with a few questions after each of the presentation briefly, so we can move to the next presenter. There is a Q&A function below, so any of the participants feel free to write any questions you may have within the Q&A function. If you click on it, you can actually write the question. I'm going to scan through them for each of the presenters in case there's any. As you are thinking about potential questions, Weizhang, I have a couple of questions for you. So obviously BDNF affects the obesity, as you mentioned, but also the neuronal compartmental model. What do you think that, how do you think that the drug in BDNF will actually be able to affect the neurological manifestation of the disease? So because it's obviously more downstream than RAI, are we missing some of the key potential function of RAI? And what is your interpretation of that?
Yeah, so based on our genetic study of overexpressing BDNF genetically, it does treat the obesity and the metabolic dysfunction associated with obesity, but surprisingly, it can also improve some of the neural functions such as social interaction, right? So it goes beyond just the obesity. So that's why in our drug trial, we are going to test all of the basically defect, behavioral or obesity we can measure in SMS mice.
Okay, thank you for the answer. And also the other point I wanted to ask you, you're now going to test at least the therapeutic intervention in adult mice, which is absolutely exciting because unfortunately there's always a misconception that SMS can only be treated as children. And actually we don't know that. There's a higher probability when you're a child probably to be treated, but it's not impossible that you may do an effect also in adults. So I really look forward to seeing your data in adult mice as well. One simple question, are you going also to assess though the effect in younger mice, either adolescents or newborns, to see if the impact on the drug may even be more substantial and more effective in a way?
So right now we are focusing on adult because these drugs are expensive and we have limited amount of resources and everything. So we want to focus on kind of the most difficult thing, because if we can treat adult mice, then we have more reason to, we will be very optimistic to treat even younger mice. So that is the one thing we're focusing on right now, but eventually we can focus on different ages and see if there's any defect, right? If there are some features cannot be rescued at adult, we can push forward younger and to see if we can treat the features in younger mice for sure.
Very good. So we got a question online from Richard Cartwright. So I'm going to ask you that question. Is there any known negative effects if the BDNF is overactivated or excessive? So
too much BDNF, I think, is also a tricky thing. So one thing we noticed was that if you had too much BDNF in wild-type mice, so remember the genetic overexpression of human BDNF in mice, we also overexpress that in mice. And as it turned out, if mice have overexpressed BDNF, their body weight is basically what we saw was fine. The adipocyte size was similar to control, but the only thing we found weird was the social interaction. They actually become more shy. So it looks like the too much or too little BDNF can modify the social interaction in wild-type mice as well.
Thank you, Weizhang. And maybe just to add to that, I would say that one of the advantages of having a small molecule as an approach is that you can modulate the dose and to be able also to assess the level of activation of BDNF. So that could be certainly the element that will be assessed, of course, in mice, and then potentially if one day we'll be able to in human as well. All right, so I suggest to move to the next presenter. Again, thank you very much, Weizhang, always very eloquent and clear. Our next presenter of the day is Jess Cardin. Right, Jess? I don't think I ever met you in person, actually. You worked together with Mike. We had a few interactions with Mike Higley in the Higley Cardin lab. So Jess is an associate professor at the Kavli Institute for Neuroscience in the Yale Department of Neuroscience. Thank you very much for joining us, and again for all the work that you are doing together with Mike. So while we know that the RAI is the central factor in SMS, we don't know how its disruption alters the development and function in the brain. So the Higley Cardin lab at Yale combines a microscope with genetically encoded sensors that light up in the brain of mice when neurons are active, and providing a readout of brain function with extremely high spatial and temporal resolution. It's actually quite impressive to see the data. With these tools, we can investigate how patterns of a neuronal signal works. Now, by imaging neurocortical signal in mice models of disrupted RI1, we hope to identify unknown functional signatures of the brain activity associated with SMS. And with that, I would like to let Jess start her presentation.
So Carol, you'll make her a host, please.
Jessica, I just made you a host. If you wouldn't mind, please hitting record. I'll get the recording from you later.
Sounds good. Let me find the record button.
Bottom,
It's usually below, and it's a red button.
Yep, got it.
Thank you very much.
Okay. So first, I really want to say thank you so much. It's really a privilege to be able to share some of the data from this project with you all. This is a project that is entirely supported by the SMS Research Foundation and would not have been possible at all without that support. And this particular project is a collaboration between my lab and my Kigley's lab at Yale. And I'm going to tell you a little bit about the methods that we're using, and then I'll show you a little bit of what that data looks like and some of our most recent results. So we're really interested, as Luca said, in phenotyping alterations in brain activity in mouse models of SMS. And I think everyone here is really invested in identifying the relationship between dysfunction of the gene REI1 and SMS. And looking at the brain, there are a lot of levels to unpack in between the gene and the phenotypes that we see in the syndrome. And that goes all the way from signaling within individual neurons in the brain to the way that neurons talk to each other through synaptic connections, the activity profile of individual neurons, and then the activity of populations of neurons as they're talking to each other during cognition and behavior, how those circuits can be organized, and in particular, the dynamics of brain activity in large networks. And one of the reasons that we're really quite focused on this level here that I'm highlighting, the network dynamics, is that we know that brain functions like learning and memory, perception, cognition, language, these all are functions of large-scale networks of interaction among different brain areas, particularly in the cerebral cortex, which is really important for cognition, for language, for perception, and those kinds of functions. And so we'd really like to focus in on how RA1 dysfunction disrupts network dynamics in the brain. Our model for this is, as in the previous talk, is a mouse model in which we are disrupting the function of either one or both copies of RAI1 using an inducible Croix system. So this is a mutation in specific populations of neurons. In this case, it's all of the excitatory principal neurons. So about 80% of the neurons across the cerebral cortex. And we are introducing an indicator to report neural activity using a method that we developed a few years ago, where we can use adeno-associated viruses to infect the whole brain all at once to express GCAMP6, which is a calcium indicator. And the reason that you would want to put a calcium indicator into the brain is that calcium is a really excellent proxy for the activity of neurons. And this allows us to read out in fluorescence how active neurons are across the brain. But we don't really, in this case, want to look at just single neurons, because we really want those whole networks that span multiple areas of the brain. So we really want to zoom out and take snapshots of as much of the, in this case, the cerebral cortex, as we can capture at once. So in this case, we're taking snapshots in real time using something called wide field or mesoscopic imaging. And we're putting mice underneath a microscope, where we have an LED to illuminate the top of the brain with light, and then we have a camera to capture fluorescence. And this is what that looks like. So this is a top-down view of a mouse's brain. This is sped up a little bit, so it's not in real time. And what you're seeing is fluorescence that indicates the activity of populations of neurons. So warmer colors indicate more brain activity. And just to orient you, these are the two sides of the mouse's brain. This is the front and this is the back. And so the nose would be up here and the tail would be down there. And the cool thing about this is that it's pretty non-invasive. We get these snapshots right through the skull of the mouse. Okay, so we have two goals with this kind of project, right? One is that we really like to understand the links between REI1's function as a gene, the patterns of brain activity that correspond to cognition or behavior or different states of the animal, right? And how that relates to some of the behavioral phenotypes that we see in SMS. But as a second stage, one of our other goals is really to develop a platform that works as a sort of moderate throughput screen. We don't just want to understand what's disrupted about brain activity. We want a way to screen for the impact of potential therapeutic strategies to rescue perturbed brain activity. And those could be gene therapy, or they could be small molecule or pharmacological manipulations. But we really want something that gives us clear phenotypes of brain activity, clear benchmarks for potential therapeutics, and we want it to be rapid. We don't want this to take years. We want to have a really, quick assay for rescue of perturbed brain activity. And one of the nice things about this technique is that it gives us rapid snapshots. It only takes about half an hour to an hour of data per mouse to get phenotypic snapshots for each animal. We can do this imaging throughout the lifetime of the animal, from development all the way through adulthood. We can do within animal comparisons across a pretty long time scale. Okay, so to give you some examples of what this looks like, so on the left-hand side, I'm going to show you data from a wild-type animal, which has 2 functional copies of REI1. And on the right, I'm going to show you a movie of brain activity from a homozygous mutant mouse in which both copies of REI1 have been lost. So on the left-hand side, you have tails on the bottom, and the left and right hemispheres of the brain, you're seeing a top-down view through the skull. And in wild-type mice, you see big patterns of activity. Again, warmer colors mean more brain activity. So this is a mouse that's sort of hanging out, going through lots of different behavioral states during wakefulness. You see lots of big patterns of activity on both sides of the brain, as well as pauses in between those big bursts of activity. And waves of activity will move between brain areas. Now, if we then compare to our homozygous mutant on the right-hand side, if you watch the activity here, you'll notice a couple of things look a little bit different. We do see some large bursts of activity, but far fewer than in the wild-type mouse on the left-hand side. And a lot of the activity that we see appears to be more diffuse and at a lower level. So here we see less of those bright yellows because the activity is a little lower. And much of the activity appears to be a little more homogeneous across brain areas as opposed to punctate and moving in discrete waves. So this kind of snapshot gives us some clues to what to look for in examining patterns of brain activity in our different mutants. So I will take you through some of the ways that we analyze this kind of data in our awake behaving mice. One of the cool things about mice is that they do a lot of the same things that humans do. So everyone who's listening to this presumably has noticed at some point that, sometimes you feel like your brain works differently at different points in the day. You have moments where you're really alert and focused and attentive, and you feel like you're able to process information quickly, form memories, retrieve information. You're really on. And then you have other moments where maybe you're a little drowsy or a little sleepy, or you're tired, you're worn out, and you're not really processing information as well. So mice also similarly go through different states of either deep quiescence and drowsiness, or moderate arousal, or very active moments where they might perform a motor action like grooming or running. So what we do is we divide our data into different behavioral states to look and see what the relationship is between activity in the brain and the different behaviors that the mouse is exhibiting. And here I'm showing you 3 different categories. One of the ways in mice and in humans actually that we categorize arousal or active states is to look at facial movements. So here I'm showing you on the left-hand column a high face state, and this is the mouse sitting still, not doing any overt motor movements, but showing lots of motor movements in his face. Humans do this too when we're alert and attentive. So on the left-hand side is a moderate arousal state of high facial movement. In the middle is grooming, which is a repetitive motor activity. And on the right-hand side is a very high arousal state where mice start to run on a running wheel. And our wild-type mice are on the top. So these are regular mice with 2 functional copies of REI1. In our middle row are our heterozygous mutants, which are missing one copy, so they have one functional copy of RAI1 left. And on our bottom row, homozygous mutants, which have no functional copies of RAI1 left. And immediately you can see that the difference between our wild type mice and our mutants is that our mutants over all these different behavioral states and different behaviors that they produce have a less activity in the cerebral cortex in this top-down view. And again, warmer colors would be spots with more neural activity, more firing of neurons. What's interesting about this data is that we appear to also have a bit of a dose-dependent effect, where our heterozygous mutants have a modest reduction in overall activity levels, but our homozygous mutants have a more substantial reduction. Importantly, this isn't because the mice are exhibiting different behaviors. In this plot on the upper right, you can see our three categories: wild types, heterozygotes, and homozygous mutants. All three groups of mice exhibit the same behaviors in about the same proportion. So there are no major differences in the behaviors of these mice. They all exhibit these three different behavioral states at about the same rate. And if we look a little more quantitatively at activity levels across the brain, we find a sort of stepwise function where our wild type mice in blue show the most neural activity, our heterozygotes in orange show less, and our homozygous mutants with no functional copies of RAI1 show the least activity. And this is true across the brain. So here I'm showing you an area in the back of the brain. This is visual cortex. An area in the middle of the brain, this is somatosensory cortex, and an area in the front of the brain in motor cortex. And across this front to back axis, we see an overall decrease in neural activity in the cortex in our RAI1 mutants. Okay, so there's one phenotype here easily seen in this kind of data. Now, the other thing that we can get from this kind of snapshot of neural activity is something called functional connectivity. And you may have heard of this in people discussing MRI data from humans. Functional connectivity is just a correlation-based measure of the relationship between activity in different parts of the brain. So how coordinated is neuronal activity in two different parts of the brain at any given moment? And again, I've divided data here into 4 different behavioral states with increasing arousal as you go to the right. And again, our wild types are on the top, our heterozygotes are in the middle, and our homozygous mutation mice are on the bottom. In each case, I'm showing you a matrix of correlation values where all the different areas of the brain are shown on each axis. And so the values that you see here are color code indicating how correlated are the patterns of activity in 2 different brain areas. Now, these plots are, I admit, kind of hard to look at. There's a lot of yellow and green and blue here. So in a moment, I'm going to show you something that's a little easier to interpret as you look at it. But just by eyeballing this, you can sort of see that there's more warm colors, higher correlations in our mutant animals. Now, if we take this kind of plot and instead calculate the difference between our control or wild type animals and our mutants, a pattern starts to emerge. So here I'm showing you the difference in the degree of functional connectivity or correlation that's shown by our heterozygous mutants on the top and our homozygous mutants on the bottom. So warmer colors here indicate enhanced functional connectivity or correlation. Cooler colors indicate a decrease in functional connectivity. Overall, across all the different behaviors that we've seen, all the different states of the animals, and in both heterozygous and homozygous mutants, we see an overall enhancement of functional connectivity. So activity is more coordinated across the cortex in both sets of mutants here. Suggesting that although there's decreased total activity seen in these animals, the coordination of activity across areas has been greatly increased. So this is sort of the end of our first pass at looking at these kinds of experiments in these animals. And what we've come to is 2 clear phenotypes in the neural data from the in vivo imaging in these animals: a decrease in overall neural activity and amplitude, and an enhancement of cross cortex functional connectivity in the mutants across multiple different behavioral states. So now we can start to think about going back into this kind of screen of neural activity to compare the activity in our mutants to that in perhaps mutants treated with either gene therapy approaches, small molecule approaches, to see whether treating animals perhaps at different age points, different points during development, gives us different outcomes in terms of amelioration or rescue of our neural phenotypes. So all the data that I just showed you was collected by a very talented graduate student named Rachel Oren. This is her main project in the 2 labs. She's co-mentored by myself and Mike Higley. And thank you very much for listening.
Excellent. Thank you very much, Jess. A round of applause virtual. Thank you for your talk. Again, for anybody, any question you may have, go to the Q&A function. As we wait, I'll start with one question. Just to know that the RAI is known to have a role also in the sleeping cycles and the activity during sleep. So is it possible to do this type of measurement as mice are actually sleeping? I don't even know if it's technically possible, but I'm wondering if that could be done and that could show if there's truly a difference in activity during sleep.
So we, in this initial project, we really focused on developing a reasonably high throughput screen. So we really wanted that fast screen, like, look at every animal for a half an hour or an hour to really process through looking at comparative approaches. But at the same time, we've been developing a parallel set of technology with a head-mounted version of this microscope. So we now have a freely moving version. So we are able to do sort of full circadian imaging in mice freely moving in home environments. So our hope is that we can also catch sleep-wake transitions and activity during sleep in the same animals. That's not a high throughput screen in the same way, but certainly our faster high throughput screen has pointed us in some really clear directions that are worth digging into with a sort of more long-term approach. Absolutely.
Wow, very fascinating. Thank you. So look forward to seeing some of that. And and we heard before about the BDNF activator. I think that's certainly the small molecule that we could test in your system. Maybe a collaboration between you and Professor Wei Zhang could be a way to go here to accelerate some of our research. All right, so I don't see any other questions. So I suggest to move if there's none. And by the way, panelists, feel free to unmute and ask questions too if you want. You're allowed to, don't worry. So we can move to the next presenters. And we have both Michael Sutton, Dr. Michael Sutton, as well as Dr. Iwase, Shigeki Iwase. I hope I pronounced your name correctly. So I'll start with Dr. Sutton. He's a professor of the Department of Molecular and Integrative Physiology and senior investigator in the Michigan Neuroscience Institute in the University of Michigan School of Medicine. And the research program in his lab focuses primarily on the establishment and refinement of synaptic connections in the developing brain and remodeling of those connections by activity in mature neural circuits. His work has been published in top scientific journals. Dr. Sutton enjoys a close collaboration with Dr. Shikage Iwase's research group in the Department of Human Genetics in Michigan. Both of them are here tonight. So together, their laboratories have launched an intensive effort to understand how chromatin remodeling contributes to neurodevelopment disorders. A significant aspect of the initiative is aimed at understanding how loss of the gene RAI alter neurons, synapses, and circuits in SMS. Their goal is to contribute to developing novel therapeutic strategies to treat SMS based on a deeper understanding of the cellular roles of RAI in the brain. With a brief introduction, I would like then Mike and Shigeki present.
I can only make one of you a host. Who would you like it to be?
Make me a host if you don't mind. We think maybe I should speak rather than us going every other word.
Okay. And once I make you host, if you wouldn't mind, if this stops recording, if you would start recording, please.
Sure
and I'll
will.
get the recording from you later.
Yep. Okay, looks like we're still good.
It looks like we're still recording.
All right, wonderful. Okay. Let's get set up here. Still recording? Wonderful. Well, thank you very much for that wonderful introduction, Gianluca. It's wonderful to meet you. Thank you, ladies and gentlemen, for being here. I'm absolutely thrilled and excited to present some of our recent work on RAA1 and how we're thinking about how this work might lead to some novel therapeutic strategies for SMS. So let me start by sort of introducing you to the team. These are the people that do all the work in the lab. These are the folks in both my lab and Dr. Iwase's lab who are focused on RAA1 or SMS. And they're a really talented group. They're really devoted to this work. They work very hard to try to get a deeper understanding of what RAA1's function in neurons is. And so we're really indebted to them. And I should say that all of the work that I'm talking about is a partnership between my lab and Dr. Shigeki Iwasa's lab in the Department of Human Genetics at University of Michigan Medical School. We're grateful for extramural funding from the National Institute of Health. This really helps support our broader interests in chromatin regulation, but we're especially grateful for support from the SMS Research Foundation, because this has allowed us to ask very targeted questions about RAA1 function and how that might relate to SMS disease progression. So the primary goal of our work is to advance the development of therapies for SMS based on a better understanding of the physiological roles for RAA1. We know that loss of RAA1 has pretty severe effects for brain development, but we don't really know a whole lot about what RAA1's roles in neurons are during normal development. And so a big goal of our work is to try and get a better understanding of that. So just in some context, you know that neurons, like all cells, have specialized functions, and this is because they express a unique complement of proteins. They express ion channels to make them excitable. They express neurotransmitters and receptors to allow them to communicate with other neurons. These specialized proteins, the blueprints for these, of course, are in the nucleus in the form of the DNA genetic code. These genes can be read out and copies made into a molecule called RNAi through a process called transcription. These RNA molecules have these coding regions, but also these intronic regions that are non-coding. These intronic regions get spliced out, and then these mature mRNAs get exported into the cytoplasm, the rest of the cell, where they serve as a blueprint to make these proteins. So what we do know about RAI1's function is that it seems to function here. Seems to function at the level of DNA and regulating the expression of various genes. Although RAA1 doesn't bind DNA directly, it does bind a complex of proteins associated with DNA that we call chromatin. These are histone proteins, and the DNA wraps around these histone proteins, and RAA1 seems to recognize this complex and thereby regulate gene expression. So given this, we want to have a deeper understanding of RAA1's function. And so to get at this, we want to profile the genes that are direct targets of RAA1. And so to do this, we take advantage of the fact that during transcription, the DNA bases in DNA get copied into an mRNA template, but thymine, the normal base for DNA, gets replaced with uracil in mRNA. So we can take advantage of that and give cells a different form of uracil called bromouridine that they then take up and incorporate into their mRNA copies. This allows us to sort of tag those newly made mRNA transcripts as the cell's making them. So we can ask, when we acutely remove RAA1 from cells, what are the consequences in terms of the genes that are being made? How does it directly regulate gene transcription? And so to get a better understanding of this, we want to study this in an additional context. We also want to know, are these RAA1 dependent genes, which we think are important possibly for disease progression, are they activity responsive or not? And so, as you know, neural activity is very important for neural circuits, but it's very important that neural activity be stable. And because of all the synaptic connections between neurons, we know that that activity has the propensity to become unstable very quickly. You can have persistent increases in activity, like seizures, but even things that are pre-seizure that compromise circuit function. But you can also have too little activity. So circuits have to maintain this stable level of activity. And how do they do this? Well, fortunately, neurons are endowed with a safety net. These are processes that kick in when activity gets too high or too low. And so when activity gets persistently high, a process kicks in first at the level of transcription that basically weakens the connections between neurons to allow this firing rate to come back into this stable range. So the signature of this event then is this downscaling of synaptic function in order to bring neural activity back into this stable range. Likewise, when activity gets too low, neurons upscale their synapses. They increase the strength of their synapses in order to bring activity back into the stable range. We know these processes are very important, particularly in developing neural circuits, for maintaining stability and allowing these circuits to function properly, even though there's large-scale synaptic remodeling that's going on. It's also important, however, that these mechanisms don't get turned on when they're not supposed to. So you wouldn't want to turn on this upscaling program, for example, when activity is stable, because this might push you into an unstable hyperactive state. And as we heard from the first talk today, there is some evidence that neurons are in this hyperactive state, at least in some regions of the brain. So to profile new genes, we took advantage of this Bruce-seq approach. We took mouse brains and we prepared neurons from them, allowed them to mature in cell culture over a few weeks, and then we disrupted activity. We either suppressed activity with an agent called prototoxin. This blocks the ability of the neurons to fire action potentials, or we took advantage of this agent bicuculin, which basically increases activity in the network by decreasing inhibition. Okay, so neurons are either persistently activity suppressed or persistently overactivated. We then profiled all these new mRNAs and asked how these states changed the transcriptional gene expression programs that the cell was engaging in. And what we found actually is that most of the genes that were turned on by activity suppression were actually not turned on during activity overactivation. So the genes were actually quite separated. So what this told us is that the gene expression programs that underlie synaptic downscaling are really separate from the gene expression programs that underlie synaptic upscaling. And so why am I telling you this? Well, the reason I'm telling you this is because we think this might really relate to something that Rh1 does in neurons. And so we got at this by first acutely removing Rh1 from neurons. We did this using a technique called RNA interference, where we could deplete Rh1 from neurons pretty acutely within about 24 hours. And we achieve about 80 to 90% loss of Rh1. So it's acute. So we can ask what happens now to gene expression when we do this. And what we find is that there's a variety of genes that are up or down regulated. Most of the genes we find that are regulated are down regulated, which is consistent with the idea that Rh1 is generally promoting the transcription of its targets. But also we find a significant cohort that are upregulated, suggesting that under some circumstances, RA1 might actually inhibit a transcription of its target genes. We found a couple features of this data set. I won't go into all of it, obviously, because the data set's complex, but we found some important features. One is that these Rh1 dependent genes, RA1 dependent genes, are enriched for synaptic proteins and regulators, suggesting that one thing that RA1 regulates are things that are important for how neurons are communicating with each other. The other big hit we found were circadian genes, which is not surprising given the disturbances in circadian rhythms and sleep-wake cycles that we see in the SMS population. So we're already sort of following up on some of these by looking at some of these downstream genes and seeing how they regulate synaptic function and synaptic phenotypes with RA1 depletion. The thing that we noticed that was very surprising is that the overwhelming majority of these genes were TTX response genes, genes that are turned on when activity is suppressed. This surprised us, and we see this at the level of the genes that are statistically regulated, the big hits, but we see this in the overall level of the transcription well. If we look and see that genes that are basically turned on by TTX, by activity suppression, generally go up when Rh1 is depleted, and genes that are turned off generally go down when Rh1 is depleted. So there seems to be a tight correlation between these genes. We don't see that correlation when we hyperactivate the neurons, suggesting that Rh1's really targeting genes that are turned on by activity suppression, the upscaling transcriptional programs. Curiously, when we actually look at this, when we suppress activity, we find that Rh1's ability to regulate gene expression is mostly lost. This suggests that it's really functioning when neural activity is stable in terms of regulating gene expression. So we used a different approach called ChIP-seq to follow up on this and to ask another question, which is, how does Rh1 bind its target genes? So can we identify true Rh1 targets, RA1 targets, by virtue of their changes in expression, but also a direct binding of RA1? And so we asked, are the genes that we identified in our earlier data, are they directly bound by RA1? And how does activity regulate this binding? And to summarize sort of a large data set and woefully small number of data panels, I just say that we do see that RA1 does bind lots of genes throughout the genome. It tends to bind in gene regulatory elements called promoters, which are the ones that are most important for regulating expression. So this is all consistent with the idea that RA1 is acting as a direct regulator of expression of those genes. The surprising thing, again, is that when we shifted activity into either inactive or overactive states, we found that almost all of this binding was gone, suggesting that changes in activity are causing RA1 to dissociate en masse from chromatin and no longer regulate its target genes. Again, most of the RA1 targets that are bound by RA1 are TTX response genes, showing that it has at least a preference for these genes that are turned on during synaptic upscaling. So what does this all mean? So we think that actually what it means is that RA1 has a very unique role in controlling the expression of these transcription programs that lead to these adaptive synaptic changes. So specifically, it plays a role in suppressing the synaptic upscaling that you would see when neural activity is stable. So under normal conditions, when neural activity is stable, you don't want to have these upscaling gene expression programs because that's going to push you into this unstable hyperactive state. But when you lose RA1, you lose that checkpoint. And now even during stable patterns of activity, you may get into this unstable state. And so we think that that's actually very, very important. So when activity shifts over either to a lower state or to a higher state, we see massive loss of RA1 from chromatin, showing that it's dissociating during these different states. So how do we test this model? So one thing we can do is measure the synaptic currents and ask, are these, if we deplete neurons of RA1, do we see that the neurons upscale? And in fact, they do. So using a technique called electrophysiology, this whole cell patch clamp electrophysiology, we can measure these synaptic currents. So these are measuring the release of a vesicle that contains neurotransmitter. In this case, it's glutamate. It binds to postsynaptic receptors. It opens up an ion channel and passes a small current, and we can measure that. And we see that these currents actually get bigger when we deplete neurons of RA1. We put our RA1 back. We can rescue this or reverse the currents back to normal. And the pattern of this increase is very reminiscent of what you see with activity suppression, in that all of the synaptic currents seem to get shifted and bigger, not just a select few. So this has, which is sort of a signature of synaptic upscaling. So together, this data suggests that one novel functional role for RA1 is to regulate this, the induction of the synaptic upscaling event, particularly when it's not appropriate for it to be induced during periods of stable patterns of activity. So to follow up on this, we're really interested in whether these roles are actually found in human neurons. So is this something that's only unique to the mouse, or can we see these same gene expression targets, the same functional role for RA1 in human neurons? And so to address this, we've taken a cellular reprogramming approach. We've taken human embryonic stem cells, used a CRISPR gene editing approach to remove either one or both copies of RA1, and then differentiated these cells into neurons, allowed these neurons to mature and form synaptic connections so that we can then measure some of these functional effects, as well as the genes that RA1 is targeting in these different circumstances. So I'm saying we're doing large-scale profiling at all these different stages of neural differentiation. We're still acquiring the data, so I don't have any of it to show you. But this includes sort of morphological analyses, as well as studying the genes, how the gene expression changes at these different stages, to get a better sense of how RA1 might be regulating neural development. But what we can actually study is test this idea of whether loss of RA1 leads to synaptic upscaling in human neurons. So in the neurons that we derive from human embryonic stem cells, loss of a single copy of RA1 does indeed lead to this upscaling effect. So we see that the synaptic currents scale up, and to a lesser extent, we see the same effect in the knockout where both copies are gone. And again, the way that this shifts, it seems like all the currents shifting get bigger, which is a characteristic signature of what we see in synaptic upscaling. So some of our ongoing projects that are inspired by this work are to, one of the main goals is to identify the genes in human neurons that are directly regulated by RA1 and are conserved. We think this is important for identifying novel molecular pathways as therapeutic targets. And this will allow us to test if modifying those pathways impacts these synaptic and behavioral effects of RA1 deficiency. So we really want to know whether the genes that are turned on in mice and turned off in mice are the same genes that are turned on and off in human neurons. And we're hopeful and even cautiously optimistic that a lot of them will be the same, but we really need to know that in order to move forward. We also want to determine if RA1 acts as a brake on homeostatic upscaling in human neurons. And if so, is this something that we can use as a potential therapeutic strategy? Can we test if reversing upscaling in these circuits, either in developing circuits or in mature circuits, whether this improves symptoms in SMS mouse models, like the ones we've heard about tonight. We're also interested in how the chromatin landscape impacts RA1 binding and dissociation. So I showed you a model of chromatin. So these are these histone proteins with DNA, and that these tails of these histone proteins can be modified by various enzymes. And this changes basically the ability of genes to be turned on and off. And so we think that in this context that these modifications are also changing how RA1 binds to its target genes. And so we want to basically determine how these different states affect RA1 binding and ask whether we can make this the states more permissive for RA binding. Because as we know, we do have one good copy of RA1 to work with. And if we can boost the function of RA1 by making this chromatin environment more permissive, this might allow some functional recovery. We can also ask whether we can bypass RA1 entirely and modify expression of specific genes by altering the landscape, again, focusing on normal downstream RA1 target genes. But this would be an approach, even if RA1 replacement didn't work, we could still target this possibly in adult circuits. And finally, we want to use cellular reprogramming to target RA1 in distinct human neuron subtypes and to look at this in patient-derived neurons. We'd like to generate neurons from patients and determine whether these phenotypes that we identify with RA1 loss are actually what we see in patients who are suffering from SMS. So let me just close by thanking again all the people that did the work, and thank you all for your attention.
Excellent. Congratulations, Mike. Those are absolutely very inspiring and very informational and educational. So I'll start with a question that we received online, again from Richard Cartwright. Is RAI something that can be synthesized? I'm ignorant on this, but curious if it is something that can be effectively absorbed, for example, a nicotine patch. Does RAI have to be delivered intravenously directly to the brain? Maybe I can quickly mention that, obviously RAI is produced by gene expression. So it's something that in theory could be synthesized. However, to bring that into the brain and absorb it is a very complex process. So certainly cannot be done through a nicotine patch. It needs to reach directly into the brain. And then, as mentioned by Mike, is the regulation and the function is largely within the nucleus. In addition to that, RAI is a very complex and large protein. So it's pharmacologically, I would say, impossible to do that through the skin or through a nicotine patch, or even intravenously it will be almost impossible to go into the brain. In addition to that, due to the high instability of the protein itself, this type of approach will not work, will be very difficult to approach. What you can do though, and that's the therapeutic intervention you heard previously, you can induce the increase of expression of the gene into the brain. You can use a gene editing and gene therapy approach, Cas9 approach, to be able to increase the gene expression. And that's one of the approaches that you heard. Or you can use a small molecule that could somewhat stabilize the expression of the gene or increase the expression of the gene, or some of the pathway, the genes that are downstream of RAI. So it's what you mentioned as an approach, unfortunately, is non-possible, too complex. But the other approaches that we discussed today in terms of gene regulation or potential small molecule to activate the gene, it's something that it could be possible. And some of the experiments that Mike and Weizhang and Jessica referred to would use potentially some of these regulators. Another question that I have actually for you, Mike, is obviously this is quite fascinating. Now, based on the knowledge and understanding of the checkpoint activity of RAI as of today, this at least suggests to me, and maybe I'm wrong, that there could be a true role of RAI throughout life, including in adulthood. So there could be a potential role in development. Therefore, you know, it's a role that, if not restored during the neuronal development, maybe there is a damage or an alteration which is not reversible. But based on what you mentioned, and if I understand correct, that was done in adult mice. So there could be a role in the adult population as well. So this gives me a little bit of a hope of thinking that if you're able to revert the activity in the adult, you might see an effect. Is that like too much of a positive of thinking at this point? Is that only speculation, or you think it's plausible that treating the adult could lead to some benefit?
So I think it's a great question, a very good question, very deep question, and something we thought a lot about. So we think that, so there's no question that to the extent that RA1's role as a homeostatic regulator, that's going to be an important role both in development and in adults. So in that, there's hope that there's an ongoing deficit that can be corrected. Now it just so happens that developing circuits are particularly vulnerable to loss of this control and loss of this instability. So that's where you're going to see probably the biggest sort of detriment of loss of RA1. But these are things that are important throughout life. We think that these stabilization mechanisms are really important even in mature circuits, and there's actually evidence for that from a variety of sources. So insofar as this is the defect, or this is a core defect, then yeah, I think that it does offer the potential that adults could respond to treatment options if we can reverse it.
Yeah, thank you.
It's a great question.
Excellent, and absolutely fascinating. So I think this is fantastic work. Thank you so much. All right, I have a last question here, which is not for you. Was actually in the beginning of the discussion from Jen Goglia. The question is, what is the average age that kids are diagnosed? I have to say, I honestly don't know if there is data today on the average diagnosis, at least in the United States. What I can tell you that is largely dependent from probably country to country, state by state, the ability to do early diagnostics and genetic testing. I think there's a huge variability across the board, I'm pretty sure. What certainly we observe is that in the past, diagnosis happened very late because obviously of the fact that genetic testing in general and genetic sequencing was much more complex, expensive, and not routinely done. Today, we can do genetic testing and sequencing for a few $100s or even less, and probably it's going to be even less expensive in the future. So I do believe that it's going to happen more and more frequent. That's why we see a lot of diagnoses now within the first 1 or 2 years of life already in SMS. All right, and with that, I think today we'll learn a lot about future options. I think this is extremely high quality research that we've seen today. I am extremely pleased about the progress. As you saw, there is a progress in understanding the underlying physiopathological mechanism of the disease. There is a better way also to measure, even see visually activity of neurons. And we even saw promising potential for therapeutic intervention, including small molecule or gene therapy with CRISPR-Cas9. So I encourage further collaboration across all these different groups. And with that, I think we can conclude the discussion today. There is a question, which is more a technical question by Ben Popper. Will the recording of this meeting be available? I assume it will be.
Yes.
But I will give it to then Jen and Chris to close it out.
Thank you. Yes, we will take care of that, and we will make it available to our audience. So Terrell, if you could put up the slides. I want to thank all the panelists for a great job and the researchers for their time. We really appreciate it. And Jen's just going to close it out by thanking some of our sponsors again and thanking for all the people that really worked hard to raise money for our foundation.
Yes, everything that Chris said, yes. And I would like to take the last few minutes because I know we have gone on way past the 8:30 mark. So those who've stayed on, thank you for staying with us. Hang on just a few more minutes as we just make some closing remarks. First, and again, I'd like to thank Vanda Pharmaceuticals for their support tonight. I would also like to thank D.E. Harvey Builders for their support, STP Nuclear Operating Company, and Living Special Needs. Without them, we couldn't have done all that we did tonight, so thank you. As you know, that this was the final piece of our fundraiser this month, Steps Move Science. So I would like to take a moment to give a big shout out to the teams that participated. Please take a look and enjoy the photos they've shared with us. Ben's Bunch, thank you. Strength for Sydney, Miller Family, Running for Research, Ensemble pour, this is my French here, Notre Hubert, Team Vanda Pharmaceuticals, Robin's Avocados, KK's Cause, Chloe's Crew, and Team Sour Patch. Thank you so much. We could not have done this without you. We appreciate for the returning families who did a team for us last year, and welcome to the new teams, and thanks for that. I also would like to take a moment and share with you this amazing video that we shared last year, but it definitely touched all of us. This is Ben Kingston. He is the son of Michael Kingston, who is one of our board members, and he has a message he'd like to share with you.
Hi, I'm Ben, and I have Smith-Magenis syndrome because of a switch on the 17th chromosome. This month, we are raising money and awareness for Smith-Magenis syndrome research. So, if you could please do 17 or something and raise some money so we can learn about it. It would help me and other people with Smith-Magenis syndrome if you did. And thank you to everyone who has already donated.
Thank you, Ben. I couldn't have said it better myself. And I suggest we all follow Ben's advice and support the SMSRF in any way you can. That was quite a bit to say, and he did an excellent job. So again, to close out this evening, again, thank you to our sponsors, thank you to our team members, and lastly, thank you to John Luca, our SAP, and the researchers. It was a true pleasure meeting all of you tonight. I have to say, I would be remiss not to say this. We started this organization 12 years ago. I was there at the beginning. Where we have grown to, what we've accomplished, to have the three of you share with us tonight is truly inspiring. I hope it's inspiring to the people who joined us tonight, and I look forward to seeing you all again next year. Thanks, everyone.
Good night. Thank you so much.
Thank you. Bye-bye.
Thank you.
Thank you.
Thank you. Bye.
Thank you very much.
Thank you.
Thank you.
Appreciate
Thank you so much.
Have a great night, and thanks for your time.
Yes. Good night.
Great meeting you finally.
Great talk. Thanks so much.
Thank you.
Take care.